The great thing about living in today's world is that we get to live longer. Unfortunately, the downside is that with older age comes a higher risk of age-related diseases such as Alzheimer's, Parkinson's, heart diseases, or cancer. In order to understand human health span, we need to get to know more about biology of aging. There is no hiding it, it's complicated. It has genetic components, it has environmental components, and there is an extensive interplay between the two. One part of the human genome that has been shown to be linked to longevity is part of chromosome 4. Because individual genes can affect lifespan, we need a simple model to study it. So what we use is a nematode worm called C. elegans.
On this plate there are thousands of worms. They're really small, about 1 millimetre long, which is why we need to use a microscope in order to study them. Their genetic makeup is really similar to our own, so studying genes in the worm that control lifespan will also tell us a lot about our own lifespan. They live for about 2 to 3 weeks, they're really easy to look after, and we can also manipulate their genes very, very easily.
Even though they're so small, we can tell the difference between a young worm and an old worm just by looking at them down a microscope. On the left is a young worm, and on the right is a much older worm. The older worm can no longer move around the plate searching for food, and you might be able to see it looks a lot sicker. As with humans, there is much variation between individuals in terms of how long they live, which means when we study aging in a worm, we need to look at a whole population of worms rather than just 1 or 2 individuals.
So you can ask, how do you measure aging in a population of worms? What we do, we start with, let's say, 100 worms, and we put each and every single one of them on a separate plate. And we check up on them every single day until they die. So we need a way to assess whether they stop moving because they are dead or because they just don't want to move anymore. So what we do, we essentially poke them with a stick. In our case, it's a platinum wire. And if they respond, that means they're alive. If they don't, we count them as dead. So this way, we can draw a graph. If we start, let's say, with 100 worms alive at day 0, we have to check every single day how many worms have died. We can draw a really nice parallel to humans in here because just as us, worms can live to different age. In our lab, we are particularly interested in genes that cause changes in how long the worms live. If they live shorter, the whole curve is shifted to the left, and if they live longer, the whole curve is shifted to the right.
The aim of research in our lab is to eventually develop drugs that will influence the genetic control of aging, and therefore delay or even prevent many of the age-related diseases in humans. So in this way, we are using the worm to reveal the secrets of a long and healthy life.